Hi everyone, my name is Kashel, and today I will be presenting my research for Talaria this year. I conducted a literature review on the effects of N4-hydroxyphenylretinamide on the treatment of acute myeloid leukemia or AML cells. I was guided by my amazing mentor Yashra and along with another Talaria scholar, Tiba Ali, in the creation of this research paper. The implications of this research paper plausibly recommend further testing of this drug in treating AML. So, the area of focus of this research project incorporates topics in clinical oncology and molecular pharmacology. Specifically, we investigated this retinoid derivative, fenretinide, or for HPR, and its effects in AML cells. A little bit on the personal engagement of this research. My family actually has quite a prevalent history of cancer, and so I wanted to know more about this disease and how to treat it on a molecular level. Furthermore, as I'm also in my senior year, I am thinking of pursuing a degree in biochemistry or biomedical sciences in the near future. Now let's get to the actual topic, shall we? First, let's talk about acute myeloid leukemia. Acute myeloid leukemia is caused by clonal expression of immature blast cells in the bone marrow. So basically, myeloid blasts, or these blast cells, are these forms of immature white blood cells that cause ineffective blood cell production or hematopoiesis. This subtype of leukemia usually occurs due to mutations on these hematopoietic genes, one of which is actually the FLT3 gene, which is integral to cell survival and proliferation. Mutations on several other hematopoietic genes, such as c-KIT, or another one called nucleophosmin-1 or NPM1, will also block its tumor suppressing properties and give rise to acute myeloid leukemia. Another one, the RAS or RAS gene family, when mutated, is also a contributing factor due to its hyperprolific properties when this gene mutates. When we treat AML, we use therapies and drugs that target specific stages in the cell cycle. One that I investigated was fenretinide, a synthetic retinoid derivative. Retinoid derivatives, basically, they behave like retinoids or vitamin A. However, fenretinide is particularly unique as its mechanisms of action involve being an inhibitor to cell growth and activating these apoptotic pathways through ceramide and reactive oxygen species or ROS generation. So why is ceramide or ROS generation important? Well, this plays a particular integral role in sphingolipid metabolism. Sphingolipids are basically this subclass of lipids with sphingoid bases as their backbone. One sphingolipid that we particularly observed here and is central to a lot of these sphingolipid pathways is ceramide. This bioeffector molecule actually mediates cell death through the activation of this enzyme called enzyme sphingomyelinase. Reactive oxygen species are also important because they cause oxidative stress in the cell, which damages proteins, lipids, and it basically abrogates cellular function, leading to cell death. Next, I would like to talk about the conclusions that we drew from the research paper. So here is a summary of all the literature reviewed. First is that apoptosis in different AML cells, when treated with fenretinide, of course, may be cell line specific. AML cells are not just one type of cell. There are many cell lines. There are many models used to study the cancer biology of acute myeloid leukemia. Here we found that despite fenretinide being able to inhibit growth in all HL60, NB4, and UN37 cell lines in a time-dependent manner, the NB4 cell line declines in cell viability at a much faster rate. Next, we also found that fenretinide can also express apoptosis-related proteins in all cell lines. For example, when they conducted their Western blot analysis test, they found the presence of caspase-3, which is one of the proteins that actually promote apoptosis or cell death. Next is its cytotoxic effects on the FLT3 ITD mutation. If you remember earlier, I said the FLT3 ITD mutation was a gene that is needed in hematopoiesis. Fenretinide actually downregulates genes associated with the FLT3 ITD AML cell. So basically, the expression of an anti-apoptotic gene BCL2 was downregulated, while expressions of pro-apoptotic genes were increased after 4-HPR treatment. So basically, there was more apoptosis going on in the fenretinide treated cells. Lastly, we talked about the NR4A1 gene. This gene is usually what we call a tumor suppressor. However, when this gene mutates, it gives rise to a AML. So we found that nuclear export plays a critical role in activating the apoptotic pathway by translocating this NR4A1 protein from the nuclei to the mitochondria. So when this happens, NR4A1 can interact with the BCL2 family, specifically the BH3 domain, which is one of the pathways to lead to apoptosis. So the BH3 domain is an apoptosis agonist here. Here is a graph reaffirming my previous points. The graph shows how cell viability decreases in a time-dependent manner in each AML cell line when treated with small doses of fenretinide. As you can see, NB4 declines much faster compared to the other cell lines, which is why it is particularly sensitive to fenretinide. In conclusion, N4-hydroxyphenylretinamide demonstrates a promising outcome for the future treatment of acute myeloid leukemia. However, for a more extensive understanding of its mechanisms, more in vivo or more organism-based or whole person-based studies should be done, because most of these literature reviews were conducted in vitro. Reflecting on my Talaria experience, I think I first had a lot of challenges in time management and making my time work with my mentor. We live in different countries. We are in opposite sides of the globes, and so making time work with my mentor was really difficult because we would be texting when it would be night for me, morning for her. So it was a bit difficult to coordinate. However, I managed to overcome this because my mentor was actually extremely strategic in plotting out the phases of the research paper. So see here, we had week 1, preliminary research, week 2, brainstorming, week 3, writing, and week 4, revisions. In the month of working on this research paper, each week we'd have a different objective and we'd go over them together in our weekly meetings. I started out this research project extremely nervous because I had no prior experience with research, and when I started with this heavy topic, I was bombarded with the amount of information. I read so much journals. However, I still think that this whole experience is a brilliant opportunity for independent study and self-enrichment. My mentor was also there every step of the way and corrected my mistakes, gave me feedback. Overall, I would recommend this program to anyone with an unwavering interest in STEM and a desire to tinker their research skills. For that, I want to say thank you first and foremost to my mentor for being amazing, to Tiba for collaborating with me in this project, and to the Talaria program, of course, for this opportunity, and to everyone in the audience.